สวัสดีค่ะ
สวัสดีครับ
Welcome back to Biotech 2021. I am Elisa Stewart.
and I am Nathapong Chitjak, and we will be the MC for Room 1 this afternoon.
In this room, we will continue listening to the invited speakers. For attendees interested in listening to the contributed oral presentations on cryopreservation, those talks are being held in room 2.
We have 4 more invited talks during this afternoon session, similar to the morning sessions. Please type any question that you have in the comment box.
Our first invited speaker of the afternoon is Dr. Bart Banis from Alliance of Biodiversity International and CIAT Belgium. Dr. Pennis obtained his PhD at the University Leuven, Belgium, where he was involved in cryopreservation and the development of embryogenic cell suspensions, protoplast culture, and techniques for genetic engineering of bananas. He has trained more than 100 researchers from all parts of the world on plant cryopreservation and developed protocols for about 50 plant species. Today, Dr. Pennis will give a presentation on cryopreservation to preserve vital tropical crops for future generations.
Hello, my name is Bart Panis. I'm working for the Alliance of Biodiversity and CIAT in Leuven, in Belgium. So we are associated with the World Banana Collection. I'm also very honored that I am invited to give a presentation in this meeting. I'm very sorry that I cannot join you in the meeting because my previous experiences with the meetings that were organized in Bangkok by Dr. Kanchit were extremely pleasant. The food was nice, the atmosphere was good. So hopefully we will meet each other in near future. Some facts why we want to cryopreserve plants. Many people are undernourished and 200 million children under 5 years of age are underweighted. And what is more, the world population is expected to reach 9 to 10 billion by 2050. Especially an enormous increase will take place in the tropical countries. So you see here on this figure that by 2050, especially Asia and Africa populations will increase enormously. And while the more colder regions, North America, Europe, the population increase remains quite stable. When we want to deal with this increasing world population, we need more food. For this, reliable and sustainable improvements in yields are needed. How can we do that? We can try to increase land cultivated, but when you see this figure here, we see that from the 1970s, the amount of land that is cultured for agriculture remains quite stable. So this is probably not an option. Even in future, we might think at a decrease of the land that is cultivated due to the climate change and the desertification. We can improve cultural practices, increase biotic and abiotic stress resistance, increase yields. But to do all these kind of things, we need a broad source of plant genetic resources. So we need to preserve them. There are about 250,000 plant species identified, and only 7,000 of them are used for food by humans. And out of the 7,000, only 3 provide more than 50% of the world's food energy intake. If we broaden that a bit to 12 crops, together with 5 animal species, this provides 75% of the world's food intake. So you see that our food is depending on quite a limited diversity of plants. So the conservation is needed. But even more important than within these species, the diversity is also decreasing because what is cultured, high-yielding cultivars, disease-resistant cultivars, and cultivars which are not used for the moment, they are often neglected and they can be lost. So conservation of the widest diversity is extremely important. How we store plant germplasm. So this is one of the typical slides that I use many times. So we have in situ conservation versus ex situ conservation. And the most used ex situ conservation protocol is seed conservation. You see here the SIAT gene bank in Colombia, but also the big backup facility in Norway, in Svalbard Global Seed Vault, where important collections are placing their backup. But what is the problem is that seed storage at -20 is not applicable to all crops. For example, you have crops that produce recalcitrant seed. Recalcitrant seed means that they are not storable at -20. Examples here are avocado, mango, mangosteen, lychee, cacao, rubber tree. So you might see that these are all tropical crops. And why seeds for tropical crops are not storable? Because the conditions are as such that when a ripe seed fall on the ground, they can readily germinate because they always have the right conditions. So they do not need to go to sleep like plants in a temperate region. There are also crops for which we like to preserve specific gene combinations, like is the case in fruit trees, potatoes, cassavas, because if we would store seeds there, we would lose the typical characteristics of these gene combinations. And of course, you have seedless crops like bananas, so seed conservation is also not an option. Therefore, we need to find other ways. So one way is to store the seeds or the embryos through cryopreservation, or alternatively store vegetative tissues. How can we store vegetative tissues? This can be done in field collections or botanical gardens. Here you see an example of the IPK potato collection in Germany. Also, you can have in vitro collections where you take out the plants from the field, sterilize them, and culture them in test tubes. So you can make big collections. If, for example, a cold room, why you use cold rooms is to slow down growth, that you do not need to subculture that much. So you save a lot of time and energy. And finally, of course, you have cryopreservation, where you store the materials at -196 degrees Celsius. So the metabolism and the chemical processes are arrested. So you can store the materials for very, very long times. I also put here DNA banks. DNA banks, of course, we cannot regenerate plants out of it, but this is a good way of preserving material for molecular biologists. What is the state of the art of plant cryopreservation? So in 2017, the feasibility study was executed to screen whether a safe backup for cryopreservation or cryopreserved material was necessary. Part of this feasibility study was a survey that was sent to 26 organizations. Out of these 26 organizations, 15 responded, and we learned that they hold together about 10,000 accessions of 30 crops in cryopreserved conditions. This is only about 16% of the total number of accessions they collectively hold for these crops. Most of the accessions are still stored in field and in vitro collections. So here you see a representation of the crops that are conserved in liquid nitrogen. The most important ones are potato, banana, mulberry, coffee, allium, strawberry, cassava, to name a few. I also have to indicate that apple, dormant bud cryopreservation is not included here because that time the institutes that are conserving big collections of dormant apple buds did not respond to the survey. What are the methods for plant cryopreservation? The two ones that are the most used in the big collections are dormant bud cryopreservation and the droplet vitrification. Just to show you where these big collections are, and the methods they use. So the biggest ones are for banana, potato, twice potato even, cassava, apple, the dormant buds of apple, citrus, mulberry, just to name a few. And these are spread all over around the world. But again, you see here the two most important methods that I used: droplet vitrification and dormant bud freezing. Of course, dormant bud freezing can only be used for those species which produce dormant buds. That's a first important factor. The second requirement is that they can be grafted. And this is the case for apple and elm and mulberry. What were now the final recommendations of the feasibility study? So the first one that was that a major initiative should be launched to accelerate the development and implementation of crop preservation. Because it was estimated that there are about 100,000 accessions that need to be crop preserved, so only 10% is currently crop preserved. And also that a backup facility should be initiated to accommodate the estimated 5 to 10,000 accessions that are already cryopreserved. In fact, it would be a bit similar to what is done for orthodox seed in the Svalbard seed vault. So collection place a backup of their accessions in another place. I will now discuss a few case studies of crops and crop species that cannot be conserved through seeds, and for we which have experience in our lab in Leuven. The first one, of course, banana. We have experience with that already for 20, 30 years. Bananas are very important. It is a staple fruit for almost 1 billion of people. It produced in more than 120 countries in the tropical regions. It is the largest fruit crop in the world, with an annual production of 158 million tons. If you compare this, it is much larger than apple with 87 million tons. And the international banana trade is also quite important. So how do we store bananas at the ITC in Leuven? So we are currently having 3 conservation methods. So we have the in vitro active collection. The word active means that from that collection material is sent all over the world as rooted plants or as multiplying shoot tips. This contains about 1,600 accessions stored in a cold room, so at 15, 16 degrees Celsius, so the material only needs to be subcultured once per year. Secondly, we have the lyophilized leaf collection, so where we have store leaf or dried leaf material in a -20 freezer. This material is often demanded by molecular biologists, which are only interested in the DNA. And thirdly, we have the cryopreserved base collection, which exists now out of 1,200 accessions almost. And additionally to that, we have an offsite backbox safety backup in Montpellier, which is about 1,000 kilometers to the south of Belgium. So in case something happens with our laboratory, and you never know, the in vitro collection, the cryopreserved collection, and the lyophilized leaf collections are destroyed, we can always go back to Montpellier and regenerate the collection again from these cryopreserved materials. We developed for this the droplet vitrification protocol, so it is based on droplet vitrification of small meristems excised from rooted plants. So this is a very labor-intensive protocol. A skilled technician can excise 15 to 20 meristem maximum per hour. The meristems, if you see here, the microscopical view is only 1 millimeter in diameter. Bigger meristems and smaller meristems will not survive cryopreservation, so it is quite important to have that exact size. But this is also why cryopreservation of banana is so expensive, because the excision of meristems is time consuming. This is the protocol that we developed for the banana shoot tips. I will not go into detail of that. You can find it elsewhere in literature. But so we have rooted plants, excision of meristems, treatment with loading solution, PVS2 solution. Put them on an aluminium piece, rapid freezing in liquid nitrogen, rapid thawing in recovery solution, and then regeneration again of plants. How many accessions do we have cryopreserved already? So we are now, as I told you, about 1,200, but this is an effort that is taking place already for 16 years. So you see the number of cryopreserved accessions is increasing year by year. Some years there was almost no cryopreservation done. That was due to lack of staff. Some years we had 3, 4, 5 people working on the cryopreservation. So, what is the situation for banana cryopreservation now? An optimized protocol is available. The regeneration rates are 60, 70, 80, sometimes 100%. The major bottleneck remains the funds, because we need staff to do the cryopreservation work. I did not say it yet, but in banana, around 40 accessions can be cryopreserved per year per technician. Of course, this includes 3 independent repetitions, but still 40 accessions is not too much. And also important is that there should be cryopreservable material. So we only cryopreserve disease-free material, and this is sometimes a bottleneck. Some of the accessions still contain viruses. Second crop I want to discuss is sweet potato. So this is work which was executed by many students, but also by my current staff, Natalia and Hans. Sweet potato is very important. It ranks 7th in the world in terms of dry matter tonnage. 92 million tons are produced per year. Largest producer is China, but also a lot of production takes place in Africa, Japan, USA and New Zealand. It is mainly vegetatively propagated and it needs warm, humid climate to grow. An example of a sweet potato collection is the big collection in SIP in Peru, in Lima. They conserve more than 6,000 accessions of sweet potato in the field, in the greenhouse, as well as in vitro and cryopreservation. So they have cryopreserved more or less 230 accessions, but they say that the conservation protocol, the cryopreservation protocol, it still needs to be optimized before it can be really used on a huge scale. I also have to mention that there are collections of sweet potato in USA, Brazil and China. The protocol is quite comparable. This is a base protocol, quite comparable to the one we developed for banana. But very important here is to distinguish the different ways of regeneration. And we can distinguish full regeneration, where we are sure that the rooted plantlet can be recovered from the cryopreserved meristem. But we sometimes observe in sweet potato hyperhydricity, or we see a shoot growing, but it stops growing at a certain moment, it does not grow any further, or only a small tip or callus growth. So this is considered as survival, but will not lead to a rooted plant. So in fact, for our purpose, it's quite useless. We need rooted plants. So very important to distinguish that. So we optimized during the past 2 years some parameters. For example, the age of the shoot tip donor plants. So this is the amount of weeks, or the number of weeks after the last subculture. The results were not very significant, but best results were obtained with 6-week-old material. What you see here also is that survival is very high, always higher than 80%, but the regeneration for the controls as well as for the cryopreserved is much lower. It's only between 30 and 40%, even sometimes 20%. indicating the difference in the previous slide. We also optimized or tried to optimize the regeneration medium, and we tested 3 regeneration medium with BA, HIRAI medium, which also contains gibberellinic acid, and then we have normal morash against hook without growth regulators. The HIRAI medium proved to be slightly better than the others, but again, no statistical differences. Then we tested what the response was of apical versus axillary shoots. And we saw that axillary shoots generally for most accessions respond better, which is good news because an in vitro plant has only one apical meristem and 3, 4, 5 axillary meristems. So there is more material to do cryopreservation. So you need less plants. Effect of pre-culture we also tested, but in our case, sugar pre-culture was not so good. It even had negative effects. And then finally, with all the parameters that we optimized, we tested 10 cultivars, which are representative for the collection in SIP in Peru. And we have observed that 7 out of the 10 accessions give regeneration rates of around 40-50% at least, sometimes even to 80%. So which is good news. There are still some accessions which only give 20, 9 or 11% regeneration. We need just some tweaking there. So what is the situation for sweet potato cryopreservation? Again, I think it's a question of funding. There needs to be a lot of staff to do that work. There are some more or less recalcitrant accessions. Maybe the protocol needs some tweaking. And also there is the virus problem with sweet potatoes. So we have to be sure that the material is virus-free, free of viruses before we execute the cryopreservation. So if you want to know more about this, we published this last year in scientific reports. Then we have cassava, also some work which was done by many students and also by Natalia. It's a tuberous crop, important staple food. In contradiction to the previous crops, it is quite resistant to drought and relatively easy to grow, and the production is quite big, 303 million tons per year. Largest producers is Nigeria, but also a lot of production takes place in Thailand, Brazil, DRC and Indonesia. So what about cassava gene banks? There are 2 major gene banks, one in CIAT in Colombia, one in ITA in Nigeria, and they hold 6, respectively 3,000 accessions. They are stored currently in the field, in the greenhouse, in vitro and through cryopreservation. This is a picture of the field gene bank in Cali in Colombia, the in vitro gene bank there, and this is the field gene bank of ITA. So they together have cryopreserved about 1,500 accessions, but they say also that the protocol needs further optimization. Like was the case in sweet potato, we should also distinguish here very clearly the postal regeneration, so shoot formation, and the survival. Here you see all kinds of survival after cryopreservation, callus formation, a small shoot that does not root, but only, of course, regeneration of shoots are important. We did also some optimization experiments. This was an experiment, for example, where we compared apical with axillary meristems, and this for 3 cultivars. So what we see is that the survival and the shoot growth is relatively good for all 3 cultivars, while further plant regeneration, what you see here, after 4 and 8 weeks, is relatively low. It is only 10 to maximum 20% depending on the cultivar. What we also observe here is that apical meristems respond somewhat better than axillary meristems. And there is one cultivar which responds the best, but still only 20%. Then we did some further research to see where the lack of regeneration is coming from, and we saw that even a crop-protected meristem is showing very low shoot regeneration. So this is the regeneration of the three cultivars without any crop protection. So this is the original meristem. These are the meristems after crop protection, and this is after subsequent crop preservation. So what would be the reason for this lack of shoot regeneration? We did some microscopical studies, and we saw that even after crop protection, some parts of the meristems are dead. In fact, you see this here. And when not the whole meristem dome is surviving, this might be the cause for the lack of plant regeneration. So for cassava, what is the situation now? Regeneration rates after crop preservation still need to be optimized. How can this be done? Maybe through optimization of a regeneration media or an optimization of the cryo protocol. Another crop that we worked with is coconut. This is work done by Hannes and by Kevin. So importance of coconut. It has a production of 62 million tons per year. It is called the tree of life because it has so many useful products, the wood, the leaves, the husks, the shell, and of course the fruit and nut is used, coconut oil, coconut milk, coconut water, copra, all important products. Major producers are Indonesia, the Philippines and India, and the propagation is done by unhusked ripe nuts. What about gene banking? So there are about 200 distinct accessions stored in field gene banks. Here you see a picture of the field gene bank in Papua New Guinea. There are also some crop preservation activities done in the past. So for example, this is a picture of experiments executed in Australia and reported in 2010. And so what they do is they excise the zygotic embryo from the nut. They do a flash drying, so ultra rapid drying, and then they expose the dried embryos to liquid nitrogen. So many institutes have performed experiments on the crop preservation of zygotic embryos, also in the Philippines, Indonesia, Australia, as I mentioned already, Brazil, Sri Lanka, France. But until now, there is no collection of crop preserved coconut material available. So we also worked with the crop preservation coconut. So we also started with zygotic embryos excised from the nut. So they are then sterilized and put in vitro. These embryos can be directly cryopreserved, as mentioned previously, or the plumules can be cryopreserved, plumules excised from the embryo, or we can cryopreserve shoot cultures. But until now, shoot cultures of coconut were not available. And this was one of our first work. So what we did was we excised the zygotic embryos, we regenerated them to a small shoot, and I will show this in the next slide. We can have multiple shoots from them through axillary shoot formation. So this is the protocol that we developed 2 years ago. So shoots are put on a culture medium containing didiazuron. We get a kind of what we call cauliflower-like meristem clumps. So these are clumps containing hundreds of meristems. These clumps can be multiplied, or they can be regenerated into shoot cultures, which can later on be regenerated and put in the greenhouse. So in fact, this was our starting material for cryopreservation, a clump of multiple meristems. So what you see here, these are two jars with these clumps of meristems. We excised small 1 millimeter pieces containing 1 to 2 meristem domes, and then we exposed them to different times of PVS2, and the vitrification solution. This is the control, so you see that of course the longer these pieces are exposed to PVS2, the lower the survival. But when we cryopreserve the same materials, we see the longer the PVS2 exposure, the higher survival, up to a certain level, because this is the toxicity level of the PVS2. And we saw that an exposure of around 2 hours was optimal for this proliferating meristem clumps. The major bottlenecks for cryopreservation of coconut, so we have some promising results on which we can work further on. The tissue culture, the development of shoots from this needs further optimization. We can have some shoots, we know they can root, we put them in the greenhouse, but the protocol is still very slow. And also the cryo protocol needs to be optimized. We have here 40% survival. The regeneration of shoots from these clumps also still needs optimization. And finally, taro, or maybe better to say edible aroids. This is a work which was done some years ago by Edwige André. So people eat it on the ground stem, the leaves. The production is about 10 million tons per year. In fact, taro is just one of the 4 species that build up the edible aroids. So besides colocasia, we have alocasia, which is called giant taro, xanthosoma, which is called tanya, and cyrtosperma, which is called swamp taro. Major producers are Nigeria, China, Ghana, Cameroon, and Papua New Guinea. And also they need a very humid environment, so they thrive best in extremely wet and flooded conditions. So what about gene banks of taro? So the largest gene bank collection of edible aroids is present in SPC, Center of Pacific Crops and Trees in Fiji. So they have an in vitro collection of about 1,200 accessions. And more than 10 years ago, in collaboration with them, we developed a cryopreservation protocol for the shoot tips of colocasia. This protocol is quite similar to the protocol of banana. The shoot tips are quite similar. The vitrification protocol is quite similar. So it's the droplet vitrification that we used. And here you see some results. So the results were very, very, very good. So we tested 18 accessions. And the post-thaw regeneration rate was 73 to 100% with an average of 88%. So which means that in fact the protocol for colocasia is ready to be used. But of course we also have the 3 other edible aroids, the alocasia, xanthosoma, and the cyrtosperma. We also executed some experiments on this protocol similar to the one of colocasia. And we tested 5 alocasia accessions that we received from Fiji. And the post-thaw regeneration was at least 50%, so which is very promising. Also very clear from this is that regeneration and survival and shoot growth are quite similar. It's not like with cassava and sweet potato where the regeneration, shoot regeneration is much lower than survival. Here it is more or less similar. We tested it also on another edible aroid, the xanthosoma. Somewhat lower results, so regeneration was about 50% till 60-70%, but still acceptable. And also on the cyrtosperma, where regeneration rates were 40 to 50%. So we can say that for taro cryopreservation, Optimized protocols are available. It will just be a question of finding the funds to put the things in liquid nitrogen. So what are the final conclusions of my talk? So until 10 years ago, cryopreservation of crops was more an academic topic. So it was used to make publications, to report. But it's only since 10, 20 years that a real cryocollection have been established that contain thousands of accessions. There are still some crops that need further optimization for cryopreservation protocols. I showed you a few, cassava, coconut, some considerable optimization is needed. Sweet potato, that's already quite good. So only for a few accessions, the regeneration protocols need to be higher. And for edible aroids and banana, the protocols are already available. The COVID situation is coming hopefully to an end. Has also proven that cryopreservation is extremely important for the conservation of these vegetatively propagated crops, especially those that are preserved in vitro banks, so where you need regular access to subculture to maintain the collection. Some of these collection came into problems because of COVID. Technical staff and lab technicians could not have access to the labs to maintain the collection. So cryopreservation could give there the solution. Okay, I would like to thank you for your attention. And so this leaves me to thank many people that have collaborated with us to come to these nice results. So first of all, my staff, Natalia, Kevin, Hans, Edwige, Bart, who all performed their experiments very well, and also the students, K Leuven master students, which took also part of their work on their shoulders. And finally, our partners, so funding organizations, that have sent us plant material, and that have supported us during all these years. If you want to know more about the feasibility study, I can show you here the link to the feasibility study, and there you will find all information. So thanks for your attention, and I'm happy to answer questions.
Thank you, Dr. Panis, for your very interesting presentation. If it's convenient, can you please unmute your microphone now, as we have a few questions from the audience in the chat box. Our first question is from Dr. Kantit Tamasiri, who asked whether you use V cryoplate and or D cryoplate in your gene bank.
Okay. Hello, everybody. Can you hear me? Okay, perfect. Interesting question. Yes, we have tried a few years ago the cryoplates, especially the V cryoplate method. In our hands, we tested it on bananas and on the sweet potato, but in our hands, it did not give better results compared to the droplet vitrification. What is more, it was more time consuming in our hands. I mean, to do the same kind of the same amount of meristems per day, we had to invest one hour and a half more time per day. So that's why we stayed with the droplet vitrification protocol, but it is as effective, and people who are very handy with it maybe might use the cryoplate method, but in our hands we have chosen to keep on using the droplet vitrification method.
Thank you, Dr. Pishat. For the second question, the second question from Dr. Vibunjan. She would like to know which technique is a good choice for maximize freezing resistance for the temperate fruit tree dormant bud in cryopreservation.
So which method is best for Dormant bud cryopreservation. Was that the question?
Yes.
Yes. So in fact, dormant bud cryopreservation, yeah, first of all, of course, you need dormant buds. So these are especially crops from temperate regions. The protocol has different steps. So first of all, you need to harvest the buds in winter. Then you do an extra acclimation, so that there is a further resistance towards cold involved. Then you dry them slowly, and then you expose them to a slow freezing rate condition. So at a slow rate 0.1 degree per hour or so, 1 degree per hour. You go down to -40. So this is then freeze dehydration. So the protocol, it's standard protocol. And then after exposure to liquid nitrogen, after storage, yeah, it's a question of gentle hydration again, and then the grafting. So you need specialists to do the grafting after the cryopreservation. So, but all these protocols are available in literature. Did this answer your question?
I think so. The third question is actually a question that I had, which is when you encounter a plant species with low regeneration rates, what are the first factors that you try to tweak to improve the protocol?
Um, Yeah, first of all, you need to see where the low regeneration rate is coming from. Is this already after the cryoprotection? In case of cassava, for example, we see that cryoprotection is already giving rise to low regeneration rates. Then we need to see, okay, what is making that cryoprotection so toxic? So yeah, okay. So there is a whole series of optimization steps. So first of all, you need to know that your material is free of bacteria. Sometimes you receive or you have materials that has this latent slow growing bacteria, and you see a very low post-star regeneration, and this is often due to this latent bacteria inside. And normally in vitro culture, they do not pose a problem, but it is after cryopreservation that this problem arises. So this is the first thing to do. You have to make sure that you are dealing with bacteria-free materials. Then you test the resistance towards PVS2. That's the first step I do. See how long can you expose the meristems to PVS2 before they die. So this is then the maximum exposure. So best to do this at 0 degrees Celsius. If then the regeneration rates are still low, what you can do is to include a pre-culture phase, for example, pre-culture the plants or the meristems on media with high sugar concentrations. Sucrose, for example, works very well. Adapt the regeneration protocols. So there are a series of steps that can be improved. So until now, this worked quite well. I think yeah, there are so many parameters that you can change that most of the species you can finally end up with a good protocol. Some species are easier than others, but yeah. But I hope that answered your question.
Yes, it's like it's a very complex process. So there's many different things that you could adjust depending on what species you're working with.
Correct, yes.
Okay, Dr. Panis, we got another question from Anurag Agrawal. In your experience, are you aware of any cases of genetic instability of cryopreservation tissue that has a morphological manifestation to impact a genotype under conservation?
Yeah. Yeah, that's also often a question that comes back. My opinion is that the major problem that can cause somaclonal variation is the in vitro phase preceding the real crop preservation. In vitro, when you add some growth regulators like cytokinins, strong cytokinins, they can induce somaclonal variations. The storage itself, the storage in liquid nitrogen, there I think that the chance to induce mutation is quite limited, although it's not existing. I mean, there is continuously a background radiation everywhere on Earth. There is radiation, so that could cause point mutations, and in case of cryopreserved material, this point mutation will not be corrected. So you can have eventually a build-up of all these mutations. But I think in practice, this rate of mutations is quite low. So in theory, there is somaclonal variation possible, but to my experience, so what we did was we already have recovered banana materials after 20 years of storage, put them in the field, and the bananas looked as good as the only liquid nitrogen for 1 hour. Also, in case of other biological materials like bull sperm or things were in liquid nitrogen for 50, 60 years. They were recovered and they also looked quite normal. So there is no indication that the cryopreservation itself will reduce somaclonal variation, but the in vitro phases pre and post cryopreservation could eventually induce it. And this has to be, of course, carefully monitored. How to monitor that? The way is, of course, planting out the materials in the field, because yeah, I'm quite critical about current methods that are often used, looking for markers, SSR markers to detect somaclonal variation. According to me, this is not very optimal. So growing out in the field is the solution.
All right, and we have time for one more quick question from Dr. Nagel, who says, or who asks, what would be the next important crop to focus on?
Oh, that's well, I think that a major challenge lays in the tropical fruit species. Like you have in Thailand, you have many marvelous fruit species which are also clonally propagated. But they pose quite some problems because they have recalcitrant seed. The tissue culture protocols are not optimized yet. A lot of blackening, a lot of problems there. So I think that there is a big challenge for the tropical fruits. That's my opinion.
Thank you very much, Dr. Panis, for your engaging talk on cryopreservation of tropical crops. All right, we're gonna move to our next speaker, which is Dr. Manuela Nagels from Genetics and Crop in Germany. Nagel studied agriculture at the University of Göttingen and did her postdoctoral degree at the University of Hohenheim, Germany. Her research project involved long-term storage of plant genetic resources, survival strategy of meristematic tissues, seeds, pollen in extreme environments, translate physiology, biochemical markers of cell viability, and molecular approaches. Today, her presentation will be on molecular mechanism during cryopreservation of potato and garlic shoot tips.
Hello, it's a great honor to talk here at the Biotech conference and also about cryopreservation. And therefore, I would like to thank you and especially the organizers for inviting me. And the big thanks goes to Kanchit. Now, today, I would like to talk about cryopreservation. And I'm responsible for the cryo and stress biology group at the Leibniz Institute for Plant Genetics and Crop Plant Research, which is located here in the middle of Germany. We house the Federal Ex situ Gene Bank for agriculture and horticultural plants and have more than 150,000 accessions in our storage rooms. Most of this material is maintained as orthodox seeds. So we can dry it down to about 15% relative humidity, and we store it then in our cold storage facilities here at -18 degrees. A small part of the collection is a clonal collection, which we maintain in the field. So there are about 2,900 accessions. And then since the beginning of the 90s, my predecessor Joachim Keller, he started to back up this material under slow growth storage conditions. And nowadays we still have a collection of 2,800 accessions. From the end of the 90s, cryopreservation methods were quite well established, and so we continue until today, so that we have at the moment about 2,200 accessions in our cryo storage. However, we follow this standard system, so the ABS system for gene bank storage, gene bank conservation, so an active and basis and then security backup. And therefore our seed material is backup at the Svalbard Global Seed Vault in one of the 3 chambers which exist there in the permafrost. And at the moment there are 54,000 accessions at the Svalbard Global Seed Vault. I had the possibility to bring some of this material to the seed vault in the last year, at the beginning of the last year, before the crisis became very dangerous. And we had the chance, together with 5 other gene banks, to discuss a long-term seed storage experiment. And among these other gene banks were the ICRISAT from India, the INIA from Portugal, Embrapa from Brazil, NRSL also from Thailand. They contribute dry seeds, and this is organized by the Nordgene, who is responsible for the management of the Global Seed Vault. And the idea is to do a 100-year seed storage experiment where we control the seed aging all 10 years to understand how the seed deterioration is going on in this vault. And the interesting part for me is here that we set up in parallel a cryostorage experiment where we maintain the seeds also under -196 degrees, that we have the chance to compare this material later on when the seed storage experiment is finished. However, as mentioned, today we like to talk about molecular mechanisms during cryopreservation of potato. And therefore, I would like to introduce you to my little figures, which is here garlic, and of course also to the person who did all this work. And this is my PhD student, Kamachi Ulagapan. She's at the moment writing her thesis, but most of the results are due to her. And then I will show you also some recent results from our potato storage, from our potato studies, where my colleague Kerstin Wolf and Marion Grübe were involved and helped me to analyze this material. So, but the first question is, why do we work on garlic cryopreservation? So, and this is quite easy to answer, because you saw we have a big field study field gene bank, and we maintain about 424 garlic accessions in the field, and they have to be replanted all 1 to 5 years, depends a bit on the conditions. So this is quite work consuming, and we have to replant them because, as you may know, garlic lost its ability to form fertile seeds in the course of its domestication, so it doesn't produce seeds. It produces a kind of vegetative tissue, which are the bulbils, which can be then used for cryopreservation. And because of that, I mentioned already my colleague Joachim Keller, he started to introduce especially a different allium species into our slow growth storage. That means we maintain them at low temperatures and low light conditions. And as he was also a breeder, of course, he was very interested in that. And so we had, in the meantime, so in the 90s, more than 700 accessions in our slow growth storage. But you see, there's a continuous drop and we nowadays only have 50 accessions left, which is in our slow growth storage. And the reason is that they accumulate over time endophytes or other degradative processes which are going on, and therefore they lose their viability in the slow growth storage. And you can overcome this mechanism by planting them again in the field and introduce them again into the in vitro storage. But this is extremely time consuming, and so we follow now today the system that we would like to cryopreserve all of them instead of putting them in the slow growth storage. So, and therefore, since the beginning of 2000, we started to establish cryopreservation methods, also in collaboration of other researchers like the Korean labs, which developed very great protocols and adapted them to us. And so that we have nowadays more than 159 accessions which are safely duplicated in cryo. And at the moment we also work on a shallot cryopreservation, the continuous shallot cryopreservation. However, so why do we work on cryopreservation? I think this is now clear for garlic. It's extremely time consuming to have that in the field and in our in vitro storage, and therefore it's just important to back up them, and cryo is the best method we can use. However, there are some challenges, and which challenges do exist, I would like to explain to you now. One of the biggest challenges is it's a monocot, and multiplication is extremely difficult. So in the field, also in vitro storage, this material was collected all over the world. This means it was adapted to different growth conditions. And also in our field, sometimes it doesn't really established great. So here it's really a challenge to multiply the material that we have enough for cryopreservation. And if you have enough, you can only use few meristems from this plant because it's a monocot. That it can be a bit different for dicots, which are a bit easier then. And we have also a great variation in our field. So there exist, for example, longicuspis types, which have just few clefts, a lot of small bulbils, or Ophioscorodon types, which have also just few clefts and bigger bulbils. Then the sativum type, which are non-bolting. So very few bulbils, but more clefts, and this is similar. We have more clefts, but no bulbils at all. So each of these different types requires also a bit different procedure for cryopreservation, and therefore this is a very challenging work. So, and in addition, you can see based on that there are differences in the regrowth potential. And overall, we conducted a cryopreservation experiment on 214 accessions. And these were different types. So we use clefts, bulbils, also in vitro plants and inflorescences. And as you can see here, regrowth varies extremely. So on the median, we could use 140 explants and had a regrowth of 38%. So not all of this is therefore regarded as safely cryopreserved, only about 150 accessions. And well, of course, we would like to introduce also those accessions into cryo here, which are at this far end, to get a safely cryopreserved material. So which challenges do exist? As you remember, it's a monocot, it's hard to multiply, and there's great variation, so that we need to apply different protocols also to this material. What might be the reason for this drop in regrowth? And there can be quite various reasons. If you have ever worked on cryopreservation, you realize there are extremely many factors which influence the regrowth. It's very complex, and probably the first are the growth conditions of this material, either in vitro or field, that depends, but the year effect is extreme. So we have differences in temperature, precipitation. There can be also mechanical damages, either by weeding or by animals. There are also insects that's different in any year, diseases and infection, and also in the field, the accumulation of endophytes. And this goes along with the genetic setup, of course. And here you have from the basis, from the principal genetics characteristics of the tissue, which plays a role, availability of protective compounds, of course, and tolerances against stresses are just few examples of that. So the genotype plays a big role. In combination, and that we assume is also very important with the cryopreservation protocol, there in the different protocols appear different stresses, and you have cold acclimation, mechanical wounding, osmotic pressure, toxicity of cryoprotectants plays an important role, then also cooling procedure, slow or fast freezing, and the light conditions afterwards. So there are also few examples which play an important role. So, and I would like to focus now on the cryostresses and the dependency on the genotype of this. And therefore, I would like to introduce you the specificity of our cryopreservation protocols for garlic. And here I would like especially show you the cryopreservation protocols for the bulbils. So here this is supposed to be a bulbil. There we prepare and excise the shoot tips out of that. And then the shoot tips go onto a 10% sucrose treatment overnight, that they have the chance to recover from this stress. And after the next day, there is a loading procedure. Here we increase the sucrose content to 41%, and then with the PBS3, we have an application of 50% sucrose and 50% glycerol. And then after this treatment, they are thrown into liquid nitrogen, and the combination of this increased sucrose content, which has the possibility to drop down the water content by the osmosis, but also at the same time it increases the viscosity of the tissue. And the glycerol has the effect that it goes into a membranes and starts to protect the membranes. And this combination of the high glucose or sucrose content, glycerol, and the quick drop into liquid nitrogen leads to a glassy state. So at this state, if we have reached this state, we avoided any ice crystallization, which is very dangerous for the regrowth afterwards. Yeah, and then we have rewarming. So when we want to gain back them to life, we have a rewarming, a washing, and a regrowth procedure where we decrease also the sucrose content. And when we look at the garlic cryopreservation protocol and look at the different regrowth potential, we have from the shoot tip preparation, that's a not huge problem. However, it might be in combination with the other stresses. So the increase in sucrose, that brings us already to 90 to 100% regrowth. To after PVS3, it's 30 to 65% regrowth. And then after liquid nitrogen, and when we start to regrow this material, we have just 0 to 65% left. So during the course of cryopreservation, the stresses become bigger, so that it reduces also the material, the cryopreservation and the regrowth potential. So what might be the reason for the drop in regrowth? Yeah, as I said, in combination, so growth conditions and genotype may play a role, but especially during cryopreservation, stress increase, that you have a drop of regrowth. And can we measure that now? Because we are interested in the response of the different genotypes. But I would like to show you at first the experiment which we conducted on one genotype, and that was simply to measure the soluble sugars which we saw. And here we compared this freshly prepared material, compared it to pre-culture, to cryoprotection, and after cryopreservation, the regrowth, 1 day of regrowth. And you can see that the sugar phosphates, here we have fructose 6-phosphate, glucose 6-phosphate, and glucose 1-phosphate. You can see that all of them increase during cryopreservation, especially pre-culture and cryoprotectant. And of course, this is not really wondering. So we increase the sugar content, and so the sucrose is metabolized, so, and it's hydrolyzed to fructose, to the sugar phosphates, so that we have a continuous increase. However, you might assume that you increase the sugar content in the shoe tips. At the same time, you could assume that energy level also increases. However, this is not going to happen. We analyzed here pyruvate and also ATP. And pyruvate is a molecule which links different processes from the glycolysis to the TCA cycle where ATP is produced. And it seems that the drop in water content also reduces the ability to produce ATP, for example. And therefore, we have here a continuous drop in the pyruvate content and also an extreme drop in the ATP content. So there's an extreme reduction of the metabolism, that the TCA cycle is slowed down, and also if pyruvate drops down, the accumulation of amino acids, for example, is also reduced. So can we measure it? We see the differences during cryopreservation extremely. So there is extreme change in the metabolites of the compounds. But now we were interested in the genotype. Is there an impact of the genotype? And here we started to think about which method we could use, and applied the microscopic method and the transcriptome study. And so we included now the genotypic setup, and it compared two genotypes. We had the AL524, it's a Orpheosclerodan type, and usually assumed to produce higher regrowth potential. So because the bulbils are easier to handle, they are a bit bigger, and compared it to the AL877, it's a Longicuspis type. This has smaller bulbils and are usually a bit lower in regrowth because the bulbils are harder to handle. In this year, which we used, we saw that the LA527, it has a lower regrowth potential. However, it can be also different. So if we use a different year, there was also a much higher regrowth of this genotype. So it seems that this is very genotype year dependent. However, in this year, we use these accessions, and our microscopic pictures are also from this year. So I would like to show you the results of that. Again, we compared the different stages during cryopreservation and did electron microscopy. And to remind you, 524 has still a lower regrowth and viability, and in freshly prepared shoe tips, you do not see any difference between those both. There are also no difference between the pre-culture, even the drop in water content you cannot really see during the preparation. This is also for the cryoprotectant. However, you see that the DNA starts to go together and to combine, so that this might be an effect of the osmotic stress, but there are not really any strong other differences to see. And after the regrowth, you definitely, that's 1 day after, you see a difference in the cytoplasm and also in the nucleus. So here it seems that the DNA is strongly degraded, whereas it seems to be recover here, or at least is comparable to the cryoprotectant. So, and as the IL-5-4 is the lower the accession with the lower regrowth, you definitely have here cells which lost the viability. So because these cells cannot probably regain function. And also here you have a bigger plastoglobuli, which usually save degradative products, and they are increased here over several pictures compared to 877. So if we look then in the transcript, as we did also a transcriptome analysis, we use the Lexogene library, which produced between 8 and 12 million reads per accession. And again, when you do a principal component analysis, you see a strong difference between these two genotypes in the first principal component, so it's 98% even. And you see that fresh material is here located at the far lower end. So that means the treatments you can see in the second principal component. However, they are a bit differently distributed. What it's hard to say what it means, but you can see here for IL-5-4, it's a bit lower here than for the pre-culture of the 877. And this is rather similar here. It's also in this range compared in this range. This is after the regrowth. So we have a stronger distribution of 5-4 than 877. Now we wanted to know, is there a difference between the genotypes? So we compared cryoprotectant versus freshly prepared and wanted to know if there are contrasting genes to understand if there is a different regulation in the way how they cryoprotect the tissue. And we have on this side here upregulated genes, and on this side here downregulated genes. And you can see in 877 there are more genes upregulated than in 5-4. And it's vice versa for the downregulation. There's for 877, it's lower amount of genes downregulated than for the 5-4. However, if we look for contrasting genes, there are only 2 which are regulated. So there seems to be not very a different regulation. They go all in the same direction. So the differences between the 2 tissue seems to be more in the production of the compounds, for example. And therefore we decided here to treat the 2 genotypes as a replicate, because that is easier then to understand for the transcript analysis. And we're sure that the transcript gave us just an idea about the treatment specificity, what's the difference between the treatments. So what's the impact of the genotype? So we don't really know on basis of the transcript analysis, and therefore we move forward to the genetic regulation between the different steps in cryopreservation. And here we compared now, we did now gene ontology analysis, and we selected those genes which has a high significant level and the fold change versus the fresh material of 2. In total, we had about 200,000 genes, and 114,000 genes were significant, and this twofold change showed then those genes. And you can see, especially when you compare pre-culture versus fresh, that are the number of differentially expressed genes, that pre-culture has facilitates more down-regulation of these genes, whereas the cryoprotection has then a lower amount of down-regulation, but then there is an increased impact of genes during 1 day of regrowth. So that's the strongest effects you can see here during cryopreservation or during the application of the cryoprotectant and the regrowth. when you compare the genes at the different other levels, there's not such a strong fold change when you have the different stages to compare. So what can you really see then? And here we compared at first cryoprotection versus fresh. And I show you now the significant Go terms, the gene ontology terms, which are differentially regulated. So they can be either up or down regulated. And what is extremely significant is that we have an impact of genes which are in the cell wall and in the membranes. And I assume this is due to the glycerol, which goes into the membranes and start to transform the membranes. And especially during the cryoprotectant, where we have this glycerol impact. Then we have also a lot of oxidoreductase, so enzyme activities, hydrolase activity with electron transfer. So there are a lot of electrons involved. And you have up to 30 Go terms which are here involved. And then there is a lot of genes involved in photosynthesis and light harvesting complex. And my assumption is that the metabolism reduces all the activities which are related to photosynthesis during this cryoprotectant stage, because that's right before the stage of cryopreservation. And when we compare then this to regrowth and fresh, you see now you have a different amounts of Go terms involved, so they're up to 300 genes. So this is the stage where the metabolism, the plant metabolism starts really to function, starts really to activate everything. So ribosomes and nucleosomes are activated. You see a lot of enzymes involved which are activated here. DNA binding, hydrolase activity, again oxidoreductase, so they are highly up or down regulated. And then here especially metabolic processes, so the organism really tries to gain back function and activates everything what's necessary. So can we see genetic regulation? Yes. Definitely between the different steps, there are different types of regulation organized and controlled, and these type of regulation had probably an effect of the final regrowth. So as we saw also, there is a lot of enzymatic activity going on and a lot of oxidation and reduction potential. We wanted to investigate these effects as well. Unfortunately, at the moment, we do not have the results for garlic, but we have some of these results for potato. And as you may know, potato is also a very important crop for us because we have more than 3,000 clonal accessions of potato. It's maintained in the field and also in vitro. And potato is important and needs to be clonally maintained because it produces heterozygous seeds, and you can only maintain the clonal plant here. And we have established the DMSO droplet freezing over the years and now switched to PBS3 droplet vitrification. And you can see here our collection, which increases over the year. At the moment, we have 1,800 potato accessions in cryo, and we want to have more than 3,000. So we continuously increase the cryo storage. Yeah, and what we do here in potato, we compare as well, we do excise as well the shoot tips, but this we can do from in vitro plants. We use the upper shoot tips, but they are easily to multiply. That's the nice thing of this procedure. Again, we apply 10% sucrose, have loading and PBS3. In this way, we can use a droplet vitrification method. Then we throw it again in liquid nitrogen to have this vitrification procedure and rewarm this and followed regrowth. However, we do also have differences in the genotype response, so that we have at first 100%, and over the cryo procedure it drops down to 40 to 90%, and then at the end we have something between 40 to 70%. So And we wanted to know what might be the difference and investigated the different enzymes. And to understand oxidative stress, we analyzed hydrogen peroxide. We have had in this way untreated control, also without any acclimation, had then a fresh material, a cryoprotectant, so we left out this pre-culture and had a regrowth, and we compared two genotypes, the Desiree and the Frühnode, which are included, and you can see that over the cryopreservation procedure here the oxidative stress drops down, because this seems to be the regulation of photosynthesis. There are not so many electrons available, so yeah, the protons also drop down during the compared to the fresh and also to the cryoprotectant, and this is rather different between the genotypes. So the Frühnode, which had a lower regrowth potential, I must tell you, had obviously more hydrogen peroxide in their tissue. And this continues, and here we wanted to understand a regulator of the glycosine antioxidants, which is involved then in the redox processes. And here we have the glutathione reductase, and this is increased during fresh, so it seems that it wants to protect the material for this first treatment, for this first mechanical stress, where we have here the prepared shoe tips, but then during the cryoprotectant treatment, it goes down, so the whole metabolism, including the enzymes, go down, and then it continues to go up again. And Frühnode is now much more higher, also not significant, but it seems to try to deal with the stresses which are in the tissue. We need to further analyze this, but this is an indicator that here a lot of redox processes going on, and this absolute values seem to play a role how the different genotypes are facing the stresses. Yeah. Is the response of enzymes comparable in the potato? So as we cannot really compare the results, however, we see strong regulation in the enzymes. So what is then the consequence probably of the differences in the transcript regulation? And here it seems to be the case that the different concentrations of stresses and also of the enzymes may play a role in the different response of the genotypes for the stress. Yeah, and what is the overall conclusion now of our study? And when we compare, or when we have also garlic and potato in the tissue, is unfortunately the main factors for differences in regrowth of various potato and garlic accessions is still elusive. So we can just speculate, because we don't really find specific genes, but we find a different concentration in the enzymes and in the hydrogen peroxide, for example, which are different between the genotypes. So it seems to be, in general, the down-regulations of a lot of genes during pre-culture and cryoprotections. They are linked to redox regulation, photosynthesis, and membrane modulation. And on the other way, during cold treatment and regrowth, there seemed to be a lot of upregulation because the metabolism wants to regain function back. And in potato, we can clearly see that there's a strong antioxidant response. This is also different between the genotype. However, definitely for the search for factors and compounds that protect the plant metabolism and the tissue is required to increase the regrowth at the end. And I hope I could give you an impression how cryopreservation affects the plant metabolism and how we are interested in these processes. We will continue the study and hope that we can publish clear results with clear conclusions in a short time. And with this, I like to thank to my colleagues, also my previous colleague, of course, Joachim Keller, Frank Blattner, Angelika Sinula, Claudia Köpnick, Kerstin Wolf, and Helena Ray, who supported the studies and helped with the experiments. Then also Markus Kuhlmann, Lothar Altschmidt, Axel Himmelbach, Sandra Drieslein, Hardy Rollecek, who helped with the different types of analysis. And then, of course, the DAAD, who supported the PhD studies, and the Society for German Cryobanks, who did support the analysis. And with this, I'd like to thank you for your attention, and thank you very much again for inviting me. And then at the end, thank you very much.
Thank you, Dr. Nagel. And I do believe you, yes, I see you now. You are here with us and happy to answer questions. So for everyone either in WebEx or viewing through live stream, feel free to type in your questions in either the live stream comment box or the WebEx chat box is fine. So to start off the Q&A, I do have a quick question about different garlic cultivars or varieties. Do they usually do well under similar cryopreservation protocols, or in your experience, do they vary widely?
So thank you for this question. In terms of potato, at the moment we have decided to go for those genotypes where we know already that it seems to be quite promising results. So we gain something between 60 and 80% cryopreservation, but we know we will come to genotypes which will be more tricky. And we see that from the in vitro plants, how they grow, and also from the specific subcultivars, let's say. So we expect that we will face some challenges, and at the moment we do not know exactly how to deal with that, but that's the reason we are working on the study. So yes, there is definitely a difference.
Okay, Dr. Nigel, we got a question from Dr. Tamasuri. What are your future plans for plant cryopreservation in terms of methods and plant species at your gene bank?
Sorry, can you repeat that? I didn't.
Dr. Tamasuri would like to know what are your future plans for plant cryopreservation in terms of methods and plant species.
So, I mean, at the moment, we are still facing that we need to do the potato cryopreservation. So that's the major target. So, but garlic is also an issue. We have 300 garlic plants, but we have more than 2,000 allium species in the garden, and not species, accessions which belong to about 200 different species. And we would like to maintain them. And I think we really need to check and to test different protocols. So that will be a real challenge in future, and what our plans are, and we need to start with.
Our next question is from Anuradha Agrawal, who asks, how many garlic shoot tips per cryovial and total number of cryovials are being cryopreserved per accession in IPK cryobank? And also, are you making safety duplicates of cryo collection in another location?
Yes. So we have, so for potato, we try to do 300 and do regrowth of 100 so that we preserve 200 shoot tips for potato. But as I mentioned, it's very difficult for garlic because usually we do not have enough material. And therefore we decided to do 100, to cryopreserve 150 and use 50 for regrowth potential. So that means at the end we store 100 shoot tips and try to split them. 50% is here, so 50 shoot tips are here in Gatersleben, and 50 will go to our backup storage at the DSMZ in Braunschweig. So that's how we handle that at the moment.
Okay, we got another question from Dr. Bart Panis. We would like to know how will your result would be used to optimize cryo protocols?
Yeah, that's a good question. Yeah, at the moment we need to check. We have some ideas. I mean, there from the Arabidopsis studies we also did, we get this some input that there seem to be also some epigenetic control, and then we're thinking about to just apply different chemicals and to see if there is some modulation. So that's our strategy at the moment, but we need to analyze them comprehensively before we go in this direction. But there are some ideas.
Thank you so much, Dr. Nagel. That's all the time that we have for questions now. And we encourage anyone with other questions to please email Dr. Nagel directly, and I'm sure she would be happy to answer them. So our next invited speaker is Dr. Jorge Canhoto from the Center for Functional Ecology, Department of Life Sciences, University of Coimbra, Portugal. Dr. Canhoto graduated in biology from the University of Coimbra, Portugal, and completed his PhD in the field of plant physiology. He has published articles and book chapters mainly in the areas of plant biotechnology, plant developmental biology, and forestry. Since 2016, he has served as chair of the Division of Plant Biotechnology and Genetic Resources of the ISHS. Today, Dr. Canhoto will give a presentation on somatic embryogenesis and other in vitro techniques towards tamarillo breeding and conservation.
Good morning, here I am again, now as a participant of the Congress, not as a ISHS member. Now I'm going to present this talk about somatic embryogenesis and other in vitro techniques towards tamarillo breeding and conservation. First, I would like to thank to Professor Kanshit Tamasiri for the invitation to present these results at this Bangkok meeting called Biotech 2021. I'm sorry, I'm not being in Bangkok. I wish to be there, but the conditions do not allow us to travel from Portugal to other countries, so I have to do this presentation from my house. I'm going to give you some details about the work in Tamarillo. First, about some characteristics of the plant, then about some techniques to clone Tamarillo in vitro, mainly about somatic embryogenesis. I will refer also some of our proteomic studies on the embryogenic system of Tamarillo. Then some aspects of conservation and the importance of conservation for Tamarillo embryogenic callus, and a few data about the induction of tetraploids, about hybridization assays, and then I will give you some information about our plans for the future concerning Tamarillo. I work at the University of Coimbra. I'm a researcher and professor at the Department of Life Sciences. The University of Coimbra, of course, is located in Coimbra, a city in the center of Portugal, and the university is located at the top of the hill, and the city is divided by the Mondego River that enters the Atlantic Ocean about 40 kilometers from here to the west. The University of Coimbra is one of the oldest universities in Europe and in the world. It dates back from the 13th century, from 1290, and is the oldest university in Portugal. The old part of the university is the old King's Palace. For many, many years, Portugal was a monarchy, now is a republic, but the university occupies now the old King's Palace of the monarchy. Our department is the Department of Life Sciences. It is located inside the Botanic Garden. The Botanic Garden is a very beautiful garden from 1772, so it was about 250 years. It has a large collection of myrtaceous and gymnosperm trees and other plants. In our department, we have the classes and the research labs, and our research center is the Center for Functional Ecology. This is the building, and this is a view of the Botanic Garden. Okay, concerning Tamarillo. Tamarillo is the common name for Solanium bataceum. So the species is Solanaceus. Solanaceus is the nightshade family, a very important taxonomic group with very interesting plants like tomato, potato, capsicum, and many others. For many years, the plant was called Cyphomandra potassium, but I think in 2005 or so, the taxonomists changed the name to Solano potassium, which is the actual name. The other common names beyond Tamarillo are tomato tree in English, tomate inglés in Portuguese, tomato del árbol in Spanish, and this name in Thai. I'm not going to say the name. Tamarillo is a small tree native of some Andine countries like Peru, Bolivia, Ecuador, Colombia, Venezuela, and from South America it spreads all over the world and is now cultivated in many countries. New Zealand, Australia, some countries here in the transition from the Indian Ocean to the Pacific Ocean, in US and in Europe. The fruit reached the European markets mainly from Colombia and New Zealand, and the price is quite high. In 1 kilo of tamarillos is about 40 euros and 14 euros, sorry, and this means about 15 US dollars and more than 500 taps, that is the Thai currency. In Portugal, Portugal is quite far away from Thailand. Is in the west extreme of the Europe, and Portugal is formed by the mainland and two groups of islands in the Atlantic Oceans, the Azores Island and the Madeira Island. Here in more detail, you have here Portugal, Spain, France, and Africa. Azores Island is between Portugal and US and Canada, and Madeira Island is a little bit southern near the Canary Islands in Spain and the African continent. In Portugal, tamarillo is mainly cultivated in Madeira and also in Azores, but in Portuguese mainland it's not quite traditional. There are some trees in gardens and public parks, but not as a culture. However, in the last years, some farmers are becoming interested in this species. Here is a view of our experimental field near Coimbra, and this is a tree of tamarillo showing different fruits, in this case orange fruits. The tree grows quite fast, and after the second year in the field it starts to produce fruits. This is in Madeira Island. This is an orchard with several trees, and these are the fruits. In this case, the fruits are red, dark red. Tamarillo fruits have a high nutritional value. They are rich in different vitamins, mainly C vitamin, minerals, and some antioxidant compounds like anthocyanins and carotenoids, and the fruits are poor in carbohydrates. This is a recent work that we carried out on the properties of the nutraceutical properties of tamarillo, and by using Raman and Ftir analysis, we found that carotenoids are quite common in the pulp, fatty acids and proteins in the seeds, mainly in the embryo, and anthocyanins in the skin of the fruits. There are different cultivars. Here we have the orange cultivar, yellow cultivar, red cultivar, and the pulp and skin and the seeds have different colors in function of the different cultivars. The fruits can be eaten fresh. In general, the orange or yellow fruits are sweeter than the red ones that are more acidic, and the juices are a reflex of the fruit. So this is the juice of orange fruits, and this is the juice of the red ones. Tamarillo is used to produce many other things, to produce liqueurs. Ponche is a quite popular liqueur in Madeira Island. Ice creams, Tamarillo with lemon is a very good combination, and you can use it in other different things. In the last years, we have been working on Tamarillo breeding using both conventional and biotechnological tools. In particular, we are trying to obtain new cultivars through hybridization, to propagate plants through in vitro culture, and to use some omics techniques to analyze somatic embryogenesis. In vitro cloning of Tamarillo is quite easy, but the plant can also be propagated by seeds. However, the farmers prefer to use cloned plants because seed-derived plants are not uniform. It can be also propagated by cuttings or by grafting, but in vitro cloning is usually used. Different techniques can be applied to the propagation of Tamarillo in vitro. Here we have the example of axillary shoot proliferation. So from selected trees, we cut the branches and put them in water in the greenhouse. They sprout, and the phytomers are used to establish the cultures in vitro. Phytomers are cultivated on a media containing Ba, basilladenine, a cytokinin, and on this condition, the shoots grow, and after 1 month, they can be rooted using IBA and auxin, and plants can be obtained and transferred to soil and then to the field, and then a new cycle can be started, or the plants can just be left in the field. Here we have plants obtained by axillary shoot proliferation after about 1 month and a half, and about 3 months after transfer to soil. Another way to propagate Tamarillo is through organogenesis. In this case, the initial plant are leaves. Leaves are obtained from shoots growing in vitro, and these shoots have been previously obtained from selected trees. So leaves are cultured on a media containing Ba and NAA, and after 3 to 4 weeks in culture, buds and shoots start to develop. They can be also rooted and transferred to soil. Finally, we are using also somatic embryogenesis to propagate Tamarillo. Somatic embryogenesis is a very useful technique of cloning. It can be also used to obtain plants after genetic transformation. In some species, synthetic seeds can be produced with somatic embryos. Somatic embryogenesis is also a good system to analyze embryo development, and in some strategies of conservation, somatic embryos are also useful. In Tamarillo, somatic embryogenesis can be achieved by one step or a two-step process. In a one-step process, we usually start from zygotic embryos, mature zygotic embryos. Of course, this is not a true cloning because zygotic embryos are different from the plants that produce them. Anyway, it is a good system to obtain a large amount of somatic embryos, and this system can be used to analyze the proteomic and genomic of this process. This type of somatic embryogenesis is obtained in a media containing the auxin NAA, usually in concentrations of 2 to 5 milligrams per liter. This is the system that we use more. It's induction starting from leaves. We cut the leaves usually in 4 parts, and these parts are cultivated in vitro on a medium containing 2,4-D or picloram, 2 strong auxins, and high amounts of sucrose. The leaves have the same origin that those used to induce organogenesis. They are obtained from shoots or plantlets growing in vitro. After 3 to 4 weeks, the leaves form callus, and this callus continues to grow, and after 10 to 12 weeks, two types of distinct two distinct types of callus can be found in the same explant: embryogenic callus that are more clear, and non-embryogenic callus that are more darker and more soft. Then the embryogenic calli can be separated from the non-embryogenic, can be subcultured for years in the same medium after subcultures of usually a month or 6 weeks. And when we want to obtain somatic embryos, the embryogenic calli are transferred to an auxin-free medium and lower levels of sucrose, and on this condition the proembryogenic masses evolves into somatic embryos and then into plantlets, and these plantlets can be transferred to soil and to the field. On this picture you can see different aspects of the plants obtained by somatic embryogenesis. This system is quite interesting because two types of kali can be obtained from the same explant, which means that they have the same genetic background, and they can be also obtained in the same culture conditions. So we can compare both types of kali in terms of proteomics, genomics, or transcriptomics or metabolomics. The embryogenic calli are formed by proembryogenic masses, and these proembryogenic masses are formed by groups of meristematic cells. The non-embryogenic lines are formed by elongated cells, very vacuolated with a peripheral nucleus, and these calli are quite soft. Both types of kali can be propagated in liquid medium as suspension cultures. In the case of the embryogenic kali, can be used to produce large amount of embryos, and in the case of non-embryogenic kali, it can be used to produce enzymes or metabolites that are interesting as secondary metabolites. We have made some comparisons between embryogenic and non-embryogenic kali in terms of proteomics, and we found strong differences between them. In the case of embryogenic kali, we found mainly proteins related with the metabolism, such as enolases or threonine synthesis, but also some heat shock and ribosomal proteins. In the case of non-embryogenic kali, we found a lot of pathogenesis related proteins, which is very interesting. More interesting was the discovery that non-embryogenic kali usually possess a protein that we called NAPTC. NAPTC stands for non-embryogenic protein from Tamarillo kali, and this protein is expressed in hormones in non-embryogenic kali. When we compare these proteins with databases available, we found that this NAPTC is belongs to a group of methyltransferases that are responsible for the methylation of RNA, especially rRNA, and they belong to a group of SPO-methyl transferase that it's called SPO-methylases. These proteins are quite common in bacteria, but they exist also in different plants such as populus, maize, rice, and many others. The protein is a peptide of 221 amino acids with a mass of 25.7 kilodaltons and an isoelectric point of 6.1. We did some research with this protein. We found that the protein is usually expressed in the cytoplasm of non-embryogenic cells, as these arrows show in these two pictures, and in embryogenic cells there is no expression of this enzyme. So the enzyme is specific of non-embryogenic cali of tamarillo. After this, we have tried to prove that our NAPTC is also a methylase. So with this protocol, we made some evaluations. We used protein extracts from embryogenic and non-embryogenic cali, and also the isolated protein produced on transformed cells of the Escherichia coli, and then the enzyme was isolated from this bacteria. And we used as donor of methyl groups the compound S-adenosine methionine, that is the usually compound in cell system to that acts as a donor of methyl groups, and as substrates we use different compounds such total RNA or ribosomic RNA or DNA. And when the NAP transferred the methyl groups from SAM to the substrate, the SAM is transformed in another compound that is called S-adenosyl homocysteine, and this compound, when it binds to a substrate, it emits a fluorescence, and this fluorescence can be detected, and this system can be used to evaluate the activity of the enzyme or the protein extracts. The results show that our NAP is able to transfer methyl groups to RNA, but not to DNA. And more recently, we found also that the protein is able to transfer methyl groups to transfer RNA. Then we obtain Arabidopsis knockout lines of for NAPTC, and we compared the mutant lines of Arabidopsis with the wild type, and we found that in the aerial part of the plants there are no differences between them, but when we analyze the root part of the plants, we found that the mutants have more airy roots, more root hair, sorry, than the wild types, and also the length of the primary root and the number of lateral roots was higher in the mutant lines than in the wild type. So taken together, these results seems to point to an interaction between NAPTC and some growth regulators, mainly ethylene and auxins, because ethylene and auxins are two important hormones for root growth and for root hair formation. This system of embryogenic and non-embryogenic cali has some problems. The main problem is that embryogenic cells are contaminated by non-embryogenic cells. So whereas non-embryogenic cali is quite uniform, embryogenic cali have pro-embryogenic masses but have also some vacuolated cells. So we have been trying to purify these systems. One method that we are trying is to isolate embryogenic protoplasts and non-embryogenic protoplasts, and then compare the two types of cells. And another system is to use fluorescence activated cell sorting to separate embryogenic from non-embryogenic cells from embryogenic cowrie. This is only a picture to show the protoplasts from leaves and from embryogenic and non-embryogenic cowrie. And the cells obtained by cell sorting, when the transcripts were compared, we found very interesting results. For example, we found that in non-embryogenic cowrie, there are genes related with the ethylene functions and auxin related genes that are more expressed than in embryogenic cowrie. So this seems to prove that NAPTC is involved with auxins and ethylene on the control of somatic embryogenesis. I don't have much time to go on this, but we are also analyzing the role of some microRNAs on sugar and phosphatometabolism and this role on somatic embryogenesis. Other way of research was to find how the long-term embryogenic cali behave, and we found that this cali maintains the ability to regenerate plants, but it is usual the occurrence of somatic anomalies in these cells. So aneuploid cells are quite common in these long-term embryogenic cali, and the same occurs with the obtained plants. So long-term callus is not a very good system to generate plants and to clone Tamarillo. To avoid this, we developed a cryopreservation protocol to maintain embryogenic cali. This was published in this book, and it is very effective because the embryogenic cryopreserved cells can be regrowth and produce somatic embryos that evolve into plantates and maintain the embryogenic capacity. Time and after these periods, the explants are put in conditions to develop, to form embryos or to form shoots, and then the plants obtained were analyzed by flow cytometry or chromosome counting in the roots to determine if tetraploids have been obtained. On this condition, and especially with oryzolene, we have obtained a great, not a great number, but around 20 tetraploid plants and some mixoploid plants. And these plants were transferred to the field. We first proved that they are in fact tetraploids, so we counted the chromosomes in the roots. The normal number of chromosomes is 24, and the tetraploids, this is a mistake, is 48 chromosomes. And flow cytometry also showed that some tetraploids were obtained. In the field, the tetraploids produced fruits, but on the contrary, that we are expecting the fruits were smaller than the fruits of the diploid plants. So this is quite a frustration, but anyway, we can use now these tetraploids and cross them with the diploids, trying to obtain triploid plants, and in some species, triploids produce parthenocarpic fruits that have no seeds and are very interesting for the consumers. So let's hope that at least we can obtain parthenocarpic fruits. We did also some hybridization assays, trying to obtain plants with new characteristics. We have crossed different genotypes. We have now F1 plants in the field, and we have young F2 plants starting ready to go to the field. And we are trying to find genotypes that are drought tolerant and cold tolerant, because these two problems are quite important in Portugal. Drought tolerance in the summer and cold tolerance in the winter. To hybridize the Tamarillo, we have first analyze the characteristics of the flowers. This is an inflorescence, and this is a flower. You can see here the anthers and the pores that open to leave the pollen and to allow pollination. Here you have the stigma, and here the style. On normal conditions in vivo, the flowers are upside down, so pollination usually occurs when the flowers are closed, so the petals completely surround the reproductive organs, the anthers and the ovary and the pistil. So in this position, the pollen leaves the anthers and is captured by the stigma, and pollination and fertilization occurs. This is a set of flowers in different stages of development. This is the ideal stage to make the crosses, is when the pollination occurs, when the flowers are still closed, and in vivo this is corresponding to this stage of development when the petals are bigger than the sepals by about 5 millimeters. We did a lot of experiments on fluorescence microscopy to determine the viability of the pollen in the stigma and growth in the style, and we found that the species is mainly autogamous, as expected in a species that pollination occurs when the flowers are closed, but cross-fertilization works well too. To finish, just to show another two species that we have been working with. One is Arbutus onedo, strawberry tree. João Martins will talk about this plant on Tuesday at 11 o'clock Bangkok time. And the other species is Feijoa sloviana, a myrtaceous plant that produces edible fruits and is also used by us to analyze somatic embryogenesis. This is our future work on Tamarillo to determine the role of ethylene on somatic embryogenesis, to analyze NEP and this relationship with auxins and ethylene, analyze how rRNA methylation inhibits somatic embryogenesis, the interaction between phosphate sucrose and microRNAs, time course experiments of NEPTC expression, and tolerance to cold and drought. This is our funding for this project on Tamarillo. These are our partners. This is our group, or our group. This is Paulo Verissimo, professor of biochemistry, that helped us with the biochemistry analysis. This is Sandra, a senior researcher of our lab, and these are all the PhD and master students. And many thanks for your attention. Good morning, here I am again, now as a participant of the Congress, not as a ISHS member. Now I'm going to present this talk about somatic embryogenesis and other in vitro techniques towards Tamarillo breeding and conservation. First, I'd like to thank to Professor Kanshit Tamasiri for the invitation to present these results
If we
at
can have
this
the host
Bangkok
please stop the
meeting
video.
called Biotech
Could the
2021.
WebEx host please end the video?
I'm sorry, I'm not.
Thank you.
Thank you very much for your interesting talk, Dr. Kanhoto. I see you are here with us. We got a
Yeah.
question.
Good morning.
Good afternoon from here.
Are you listening me?
Yes. We got a first question from Dr. Tamasiri. He would like to know how about the Tamarillo fruit yield in your country? And the second question is, do you get some outstanding cultivar? And he is not sure why it is not popular to go in Thailand.
In Portugal, if I understood well the question, in Portugal the fruit is grow mainly in the islands of Portugal, in Madeira Island. And in the mainland, it's not so popular because the fruit is a little bit sensitive to low temperatures, and during frosts in spring, in some areas of Portugal, the production can be lost. So it's not so cultured like in the Atlantic Islands. I didn't get the question about Thailand. Could you repeat, please?
Oh, he doesn't know why it is not popular to grow in Thailand.
Not so much a question, just a
Yeah,
comment.
I don't know.
just a comment.
Maybe Thailand has a good climate to Tamarillo growth, but I don't know why it's not popular in Thailand.
Probably just most people here don't know about it. So you'll have to bring the tamarillos to Thailand and introduce it to
Okay.
more people around
Maybe
here.
I have to send some seeds to you to
Perfect.
spread the culture here in Thailand.
Yes, thank you. I do have another quick question. At the beginning of your talk, you mentioned several different techniques that can be used with Tamarillo. And I was wondering, are they all relatively similar in terms of propagation rates, or in your experience, which technique is the most successful?
Well, the most successful is the propagation from axillary shoot proliferation, but somatic embryogenesis works well too. The bad one is organogenesis. Organogenesis does not work very well in Tamarillo. We can induce it, but the number of plants that we can obtain is reduced when compared with somatic embryogenesis or axillary shoot proliferation.
Okay, we got another question from Dr. Nigel. Um, Dr. Nigel mentioned that we also see post-transcriptional regulation after PVS3. Have you considered bisulfite sequencing to analyze to target RNA of your RNA methyltransferase?
No, we have been working mostly with the proteins, and we have sequenced the protein, and you know the composition in amino acids, but we haven't do that yet.
I think that's all the time we have for questions for now, since we are running quite a bit behind schedule. But
Okay.
thank you so much for your
Thank you very
presentation
much.
and answers.
Bye-bye.
Thank you very much. Last but not least, our final invited speaker is Dr. Hugh Pritchard from Royal Botanic Garden, Kew, the United Kingdom. Dr. Pritchard's work research focused on the seed trade of value to conservation and sustainable use, spanning fundamental and applied science. He has contributed directly to the delivery of Kew Millennium Seed Bank project and to the development of germplasm bank of wild species of China at Kunming Institute of Botany. He has been elected as a fellow of 5 organizations and academies, is an honorary professor of the University of Sussex, and a full professor of the Chinese Academy of Science. Today, Dr. Prashad will give a presentation on seed omics and the conservation of plant biodiversity.
Thank you very much for the invitation to speak at this ISHS Congress, the 9th International Scientific and Practical Conference on biotechnology as an instrument for plant biodiversity conservation. I'm Hugh Pritchard from the Royal Botanic Gardens, Kew. I also support a laboratory at Kunming Institute of Botany, and my co-authors are Lin Liang and Chen Hong Yin at KIB, and Danny Ballesteros and Anne Vischer at Kew. The title is broad, Seed Omics and the Conservation of Plant Biodiversity. When we think about the preservation of species ex situ, we immediately think of the approach taken by the seed banks of the world, using low relative humidity to pre-dry the seed samples. The samples are then packaged with the dryness sealed in and then transferred for storage at -20 degrees centigrade. And two great examples are shown here. On the left-hand side, the Millennium Seed Bank Partnership for wild species, which has preserved 2.35 billion seeds from 80,000 seed samples and covering 42,000 species, the most biodiverse postcode or zip code in the world. And slightly in contrast, with a narrower range of species, but a huge number of seed samples, more than 1 million, backing up 87 gene banks globally and 66 countries, is the Svalbard Global Seed Vault for crops. This approach of ex situ seed banking is extremely successful. However, it can't be applied to all material. And some examples of species to which conventional seed banking can't be applied are shown here. We have co-plots of germination or viability against the moisture content of the seeds. In fact, for 10 species growing in Africa, 9 are native and 1 introduced. And as we move from the right-hand side to the left-hand side of the data sets, you see that there are 7 species that retain seed viability on drying to 3 to 7% moisture content. That's in equilibration with 15% relative humidity. And those seeds then can be quite safely transferred to -20 degrees centigrade for storage. And we call those seeds orthodox in their storage response. However, on the right-hand side, there are 3 more species, which we describe as recalcitrant. And in this instance, as you decrease the moisture content below about 40%, there's quite a rapid fall off in viability. And the examples shown are shea butter, Vitelera paradoxa, and the Sapataceae, the second most important oil seed in Africa after oil palm. Secondly, in the Myrtaceae sagigium humani, the Java plum, high in vitamin C, and the bark of the species can also be used to extract a dye. And the third species lower down is the Natal mahogany, Trichilia emetica, in the Meliaceae. There are some familiar characteristics of these 3 seeds, that they are generally large, 2 to 3 centimeters across. And as I said, they lose their viability on drying. It's a typical recalcitrant seed response. Now, if we were to transfer these seeds at high moisture content, such that we still have high viability as well, to sub-zero temperatures, then we will simply induce ice formation. And so one of the ambitions for conservationists in handling this type of recalcitrant material is, can we find a way to partially dry, but rapidly, towards the left-hand margin of the blue area, in other words, to around 25% moisture content at this lower limit for ice formation, and then transfer the material to sub-zero temperatures for storage. Now, this is an important challenge because the frequency of desiccation intolerant or desiccation sensitive or recalcitrant seeds in the global flora is high in particular groupings of plants. So in the red box on the left side, we have the IUCN red list of species, those that are critically endangered, CR, endangered EN, or vulnerable. And of the 10,000 species assessed here, either from existing data or predictions of desiccation sensitivity, you can see that the global target set within the global strategy for plant conservation, okay, this is through to 2020, but that target will be extended through to 2030, of 75% of threatened species being preserved in ex situ collections and many available for restoration, will not be achievable, and was indeed not achieved, because the likely proportion of recalcitrant seeds in those groups of critically endangered, endangered, and vulnerable species is too high, close to 40%. And a similar situation occurs when we consider the purple area here for global trees. Out of nearly 60,000 trees worldwide, it's thought again that 35 to 40% may well be producing desiccation sensitive seeds. So their conservation is a deep concern to us at the threatened species level and at the global trees level. Now, more widely, we can find these challenges of recalcitrant seeds in many, many woody and tree species of significant value. And I'm showing here from the left to the right that the red area is of increasing desiccation sensitivity. So on the left side, with tolerance of drying to about 10% moisture content, we have important beverages, tea and coffee. Then we have a number of citrus species that have variable desiccation tolerances between probably 10 and 15% moisture content. And then we have the more recalcitrant species of castania, the chestnuts, of temperate, warm, temperate lands, which are available as for food or indeed wood. Then the hugely important commercial timbers of temperate forests, quercus, the oaks, and of Southeast Asian forests, and Asian forests, the Dipterocarpus chirea species, for example, sal, and then the really highly desiccation sensitive species on the right side for food, of course, Theobroma cacao, and the important mangrove species could be Abyssinia or many other species in that group. There are 70 mangrove species worldwide. So for very many reasons, we need to find ways to preserve recalcitrant seeds of woody and tree species. One of the best ways of thinking of the design of a preservation methodology for seeds or any material from a plant is to think about the glass transition temperature, TG, and this is plotted here against the moisture content of the sample. It happens to be the embryo of a maize seed. This is the work of Williams and Leopold, which I've modified. And what we know is that as you decrease moisture and decrease temperature, then you also reduce the rate of aging of a sample, a biological sample. And for the preservation of orthodox seeds, we place the seeds in a room at 15% relative humidity, and we enforce the glassy state as the seed dries to 5% moisture content. The glassy state, which has reduced molecular freedom, then is reinforced by cooling to minus 20 degrees centigrade. And the vast majority of seed samples that are able to tolerate this treatment influence, you can see that between around 0 degrees centigrade and minus 60 degrees centigrade, there is a considerable risk of ice formation. And to protect against that, we may add a cryoprotectant to reduce both nucleation and ice growth. So I'm going to go through 3 case studies today. The first one relates to the sensitivity of seed material to dehydration. The second one to the use of cells in a cryopreservation procedure. And then the third one around omics is also to do with the germination of samples after they've been stored. So 2 examples on storage itself, one drying and cooling, and the third one on the emergence, the growth of material after storage. So the first study is on the lipidomics of seed drying tolerance. And in this instance, we were interested in the role of phosphatidic acid because it is both a downstream and upstream signal molecule for stress responses. In this instance, upstream is the production of ROS, influencing phospholipase D delta, or ABA influencing phospholipase D alpha 1 enzymes, which generates ultimately phosphatidic acid, and downstream from PA production is also a reactive oxygen species role. So it's a highly significant molecule, PA, in our understanding of the response of seed material to drying. You see here a schematic for the effects of phospholipase D in catalyzing the breakdown of phospholipids, the removal of the head group, particularly of phosphatidylcholine, PC, and phosphatidylethanolamine, PE, to produce phosphatidic acid. And the thing about PA is that it can have a detrimental effect on membrane function. So in the example shown here, similar to before, we've taken a broad range of species of varying desiccation tolerances. Now on the left-hand side, Brassica pekinensis and Pisum sativum and Cucumis sativa with Arabidopsis thaliana, there's a very high level of desiccation tolerance, so they are basically orthodox. We've then taken an intermediate species, so tolerance of partial drying, Camellia sinensis, where you can dry to around 15% moisture content, and then the truly recalcitrant seeds of Castania mollissima, Aerobotra, and Lychee chinensis. So we've chosen a broad range of species, a broad range of families, Brassicaceae, Fabaceae, Cucurbitaceae, Teaceae, Fagaceae, Rosaceae, and Sapindaceae, and a broad range of desiccation sensitivities in order to look at the role of phosphatidic acid in seed desiccation tolerance. And what we've done is extract the phospholipids and look at the classes of lipids on the right-hand side, and also the total acyl carbons in ascending order within each of the classes. And we've done that for the orthodox and recalcitrant axes and orthodox and recalcitrant cotyledons. So the left-hand data set is for P, Pisum sativum, which is truly orthodox for the axis, compared with Castania mollissima, embryonic axis. And you can see that as desiccation progresses through 2, 5, and 10 and 21 days, the first circle in the middle of the slide shows that for the recalcitrant Castania mollissima, there's a significant increase in phosphatidic acid as a result of the drying response. Now, it's not that there is no phosphatidic acid present in Pisum sativum. There is some present, but it doesn't change in its quantity. And then similarly on the right-hand side, for the orthodox and recalcitrant cotyledons, for P and chestnut, you see a similar response. The PA increases massively after 2 days of drying in the cotyledon, whereas the level of PA in the Pisum sativum cotyledon remains constant. when we compare the results amongst the 7 species from the 7 families with their varying desiccation sensitivities, then we see the sigmoidal response with seeds that retain high germination through all desiccation treatments, having PA quantities as a percentage of total lipid less than, say, 6%. And then for the more desiccation sensitive species of Castanea mollissima, Areobotra japonica, Lychee chinensis, and Camellia sinensis, you see that there is an increase in PA percentage consistent with a decrease in germination. And so for those seeds which accumulate high quantities of PA, basically above 8%, these are the ones which are most sensitive to dehydration. So we have a sort of universal marker here for the stress levels within seeds of different desiccation tolerances with respect to the accumulation of phosphoric acid. Now, the way that this works is that phospholipase D-alpha is mainly responsible for the production of phosphatidic acid. And our results showed that PLD is translocated from the cytoplasm to the membranes, where it triggers the production of PA. PA then has a negative effect, for example, on the curvature of the membrane, and increases the likelihood of leakage, holes appearing in the membrane. And to validate whether that was the main mechanism by which accumulating levels of PA had a negative impact on seed quality, we looked at some mutants of Arabidopsis. So we looked to suppress phospholipase D, particularly alpha 1, during stress. And for the stress in this instance, we hydrate an orthodox seed for 2 days. So we push it towards germination, and then dehydrate it for 5 days, and do that through a number of cycles, 5 or 6 times. And you can see here on the right-hand side that the phospholipase D delta deficient mutant of Arabidopsis thaliana shows an increasing level of desiccation sensitivity as you go through each of these cycles, but the mutant shows no effect on that response compared to the wild type. Whereas in figure A on the left-hand side, you can see that the phospholipase D alpha 1 deficient Arabidopsis thaliana seed lots have indeed a greater level of desiccation tolerance. That's the upper line, the closed circles, compared to the wild type, which are the closed squares. So we've good evidence that not only is phospholipase D alpha 1 important in the production of PA, but that the accumulation of PA in seeds of increasing desiccation sensitivity is a cause of or contributor to viability loss. So in summary, desiccation stress results in phospholipase D alpha 1 translocation to the membrane where PA accumulates following the hydrolysis primarily of PC and PE, and the overaccumulation of PA in desiccation sensitive seeds leads to viability loss, probably relating to some sort of phase separation and just destabilization of the membrane layer. And when we inhibit phospholipase D alpha 1 activity, we can reduce that PA accumulation and basically improve the survival of recalcitrant seed tissues. And I refer you to our work in plant cell and environment. The second example or case study I want to cover is lipidomics of cryotolerance. And in this instance, we've selected Magnolia officinalis as the model system for cryopreservation. Magnolia is an important family, but also an important genus with a number of species threatened with extinction. This particular species, officinalis, is a traditional Chinese medicine. The bark is used for a product which can reduce anxiety, so it relaxes you. But as with all magnolia, there's a certain level of risk of extinction. And it's known that the seed storage of magnolia is variable in terms of the tolerance of seeds at low moisture content being stored at -20 degrees centigrade. So it's important to think about new methods for the preservation of this species. So the embryogenic cells provide us with some advantages over using shoot tips and meristems. For example, we can control quite well the physiological state of the cells through precise control of the culture environment. We've used then viability detection via confocal microscopy, so that's very efficient. And as I said before, this approach is hopefully valuable for other species with non-bankable seeds. So there are more than one concept actually of non-bankable species, and what we need to do is consider the options. Currently working with Valerie Pence over the definition of exceptional species, and we've decided that there are four steps in the banking process, each of which poses problems potentially for plant conservation. Obviously not for orthodox seeds, but firstly there are too few seeds. So with inbreeding depression, for example, if there are too few individuals in the population, then seed production may be extremely limited, so you can't harvest seeds for banking. Secondly, of course, I've mentioned already recalcitrant seeds or desiccation sensitive seeds. Then the third step, even if the seed is tolerant of drying, it may store badly at -20 degrees centigrade. And then finally, there are some seeds which are extremely difficult to germinate, and you need to use in vitro techniques to embryo rescue. Now, we're including all of those four steps under the sort of exceptional species banner, but also included in here would be plant species with extremely small populations, PSESP, which has been a concept that's been worked up and worked on in China for the last decade or so. So we have to do something special with this type of material. And usually, apart from taking embryonic axes for recalcitrant seeds, we would revert to using embryogenic cells or indeed shoot tips which may be encapsulated. And the approach there is, of course, partial drying perhaps, and certainly cryoprotection, maybe using plant vitrification solution 2, PVS2, which includes 30% glycerol, 50% DMSO, 50% ethylene glycol, and also 0.4 molar sucrose. So 4 cryoprotectants, a mix of penetrating and non-penetrating cryoprotectants to help us transfer those samples in that critical zone between 0 and -60 degrees centigrade, when the material is still partially hydrated and without triggering ice formation. Now, Valerie has done that with a very rare and species that produces very few seeds, Todson's pennyroyal in the Lamiaceae. In this instance, the seeds were germinated in vitro, shoot tips isolated, and then cryopreserved using PVS2. And the approach you can see on the right-hand side being successful is ED, encapsulation dehydration of the shoot tips, or EV, encapsulation vitrification, so the cryoprotectant is permeated into the alginate bead, with 44% cryo survival. But the best survival is in using droplet vitrification, a small drop of the vitrification solution within which the shoot tip is present, with 72% cryo success after deep cooling with liquid nitrogen. So exceptional species with too few seeds, there are ways forward in terms of cryopreservation. But what of the embryogenic cells of Magnolia officinalis and our lipidomics approach? You see at the top of the slide here the different steps in the whole cryopreservation process. It's quite complicated. So there's an initial osmotic balance in the growth of the tissue in vitro, but then we apply a mild osmotic stress at step 2. Then the introduction of PVS2, which induces severe chemical drying, and then the transfer of that sample to liquid nitrogen storage, step 4, then the rewarming, 5, and then the unloading or dilution of the cryoprotectant, and then the subsequent regrowth in step 7. And if we look just below that main scheme, you can see the composition of the membrane bilayer in terms of phosphatidylcholine or phosphatidic acid. And then the data set at the bottom shows us exactly what happens to the total amount of lipid in the embryogenic cells. So at the first step, where there's osmotic balance of the cells in vitro, there's a ratio of 5:3 of PC and PE to PA. And then as we apply a mild osmotic stress as a preempting of the severe chemical drying, you see that there's an immediate decrease in PC and PE and an increase in PA. And that trigger of PA as a signal prepares the cells for the oncoming further stress, which is induced by severe chemical drying using PVS2. At that stage, during the PVS2 treatment, even though it's only over a matter of 30 minutes, there's an adjustment in the total lipids in the embryogenic cells. There's an increase in PC and PE, and there's a maintenance of PA, so it doesn't get to critical levels. And then as we go through cooling and storage, there's very little effect of those steps, including at dilution, in fact, until at regrowth, ultimately, we have a rebalancing just about of the ratio of PC and PE to PA of 6:4 compared to 5:3 at the start. So the lipidomic response of these embryonic cells through the cryopreservation procedure is such that PA is triggered as a signal under mild osmotic stress that prepares the cells to adjust, accumulate some lipids, because without the cryoprotectant, there's massive loss of lipid during this particular method, and then there's a return to equilibrium by the end of the dilution and regrowth phase. So in summary for case 2 then, with the embryogenic cultures for Magnolia officinalis, the osmotic treatment triggers PA production, it's a signal. The cryoprotectant controls against the accumulation of too much PA, which would be damaging, but you also see an increase in lipid content, PC and PE, and then at unloading and regrowth, there's a further rebalance of membrane phospholipids, and as a consequence, we see high levels of survival of these embryogenic cells through the cryopreservation procedure. Now, in introducing the omics to our consideration of preservation of biodiversity, I think we're moving far beyond what we would consider the traditional approach to cryopreservation. And we've introduced a new term, which you see in papers now, called cryobiotechnology. I defined that in 2018 as the use of modern technologies to understand the response of biological systems to low temperature cryo environments, whether natural or imposed, and leading to the production of knowledge, goods and services, including the preservation of cells and tissues for use by industry, agriculture, medical science, and conservation agencies. So the cryopreservation components, cryobanking, cryotherapy, design of D plate and V plate for cryo, is the applied end of cryobiotechnology, its design and application. It's based on an understanding of two things: fundamental biotechnologies, the omics, as I've described just now, understandings of structural biology, in vitro model systems, but also natural processes, how cells and natural environments adapt to stresses, cold or drought, their resilience processes, and also their ability to supercool. So we believe cryobiotechnology far better describes our new approaches to the preservation of plant biodiversity going into the future. The final example I want to cover is around germination, the omics of germination of a rare species. In this instance, Pseudophoenix ekmanii in the Aracacea, in the palms, critically endangered in the Dominican Republic. And for this study, we had an extremely small number of seeds. It's a type of study that we wouldn't have been able to do even 10 years ago, 5 years ago. It's a species that grows and disperses seeds at around 30 degrees centigrade in September of each year, and the lowest temperature of the environment within which the seeds would be exposed and might germinate is around 20 degrees. We decided to look at the germination performance of this rare species at 20 degrees and 30 degrees centigrade. And you can see in the upper data set here that the germination progress curves at 30 degrees centigrade are onset around 14 days. Germination reaches 80% in total, whereas at 20 degrees centigrade, it's a slower process. Some seeds start to germinate at around 1 month, but only 20% in total will emerge. And in the lower data set, you can see the effects of time on water uptake in the germination test. And there's pretty much a leveling off of imbibition, slightly different total levels, but a leveling off by around 14 days for both 20 degrees and 30 degrees centigrade. And so our focus here was the transcriptomic patterns associated with germination at 30 degrees and 20 degrees centigrade, with a comparison between 14 days when the decision is taken whether to germinate quickly or not, and 9 days when the decision would be being considered. And this is a result for the transcriptome at 14 days versus 9 days at 30 degrees centigrade for the upregulated genes. And what we find is that the decision to germinate in the embryo results in an enrichment of genes associated with plant organ, root and shoot development, which is broadly indicative of the start of embryo growth and imminent germinations. And there are many genes associated with proteins localized in the cell wall, because cell wall loosening is part of that germination process, with the membrane also. And so the differentially expressed gene sets that were regulated were 4,048. So enrichment of genes associated with plant organ development. And then for the transcriptome results for upregulated genes at 14 days compared to 9 days at 20 degrees centigrade, we have an enrichment associated with physiological dormancy and inhibition of germination, with the differentially expressed gene sets totaling 5,754. And what we see accumulating is the DELA-like protein, RGL1-like, which is a negative regulator of the gibberellin response. TRAB1, ABI5, which mediates abscisic acid-induced transcription, for example. And so many genes associated with either the induction of dormancy or the inhibition of germination. Now, what I think is exciting about this study is that we found extant homology with the oil palm genome, which I'll come back to later, Elias guineensis. So to summarize the findings of case study 3, we were looking for and found some evidence for secondary dormancy at suboptimal temperatures in the embryos of Pseudophoenix ekmanii. As the seeds slowly germinate at 20 degrees centigrade, we found enrichment of genes associated with that physiological dormancy. But the level of dormancy that's induced is incredibly shallow, because when the seeds at 20 degrees centigrade that were not germinating were transferred to 30 degrees centigrade, then another 40% of germination was recovered. And therefore, we will continue our studies in order to dissect secondary dormancy from germination inhibition as physiological responses in this species and other species. You can find our work published in Plant Biology. Now, just a few words at the end to talk about the future of omics and plant biodiversity conservation, because I think we're entering a very exciting time in the life sciences. And you can see here for, in orange, the number of new genomes which are now being published annually. It's roughly 140 compared to just a decade ago, less than 20. And overall, we have 600 complete plant genome assemblies. And that is increasingly influencing the sort of work that we do, not just in plant breeding, but in conservation studies as well. But overall, still the coverage in the plant kingdom of 353,000 species is such that we have genome assemblies for less than a third of the orders, less than a sixth of the families, less than 1/70th of the genera, and about 0.1% of known plant species have just one published genome. But I just want to return to the success that we had in transcriptomics with the rare palm, and show you how closely related the model genome that we used, Elias guineatis oil palm, was to the threatened species Pseudophoenix. So they're in adjacent subfamilies, the Aricoideae for oil palm and the Ceroxyloideae for the Pseudophoenix. And this just gives me a great deal of hope that we will see many, many more investigations using omics to elucidate mechanisms of survival, of tolerance, or of failure even, but that will certainly help us to design and improve our basic approaches to the conservation of plant biodiversity in the future. Just a few words at the end of acknowledgements to our sponsors for the lipidomics work on seed desiccation tolerance and cryoembryogenic cell cultures. Thanks to the National Natural Science Foundation of China, and for our Pseudophoenix study, our seed collecting collaborators in the Dominican Republic, and also in the UK, the Royal Botanic Gardens Kew receives granting aid from DEFRA, and our project was funded by the Garfield Weston Foundation. Thank you very much for listening.
Thank you, Dr. Pritchard. I would like to start off the Q&A session by asking, it seems like you've worked with many critically endangered species. Are there any common challenges that they share in terms of developing cryopreservation protocols, and any thoughts on how we can overcome these challenges?
Hello again, Alyssa. Nice to see you, and Nathafong also. I'm sorry not to be with you. The real challenge, I think, with the threatened species is usually access to material. Having insufficient seeds or whatever the model system is that you develop from that material to design experiments that help you take forward the preservation protocols. Of course, the best way to do that is working with a broad range of partners in different countries. So I think that's the main issue, and being sure that you're compliant with all of the CBD and CITES regulations around access to material for these collaborative programs. Sorry, did you lose me then?
No, we heard your answer. Thank you so much. I think Natapona is preparing the second question for you.
That's fine. Thank you.
Okay, we got the second question from Dr. Thamasiri. He would like to know, how is your work on palm pollen conservation?
On palm pollen?
Yes, palm pollen conservation.
We haven't worked on palm pollen, but we have been looking more recently at some threatened species in the Orchidaceae, so another monocot. And we certainly think that being more flexible in our approach to the material that we use for preservation is very important. I mentioned before about the challenges with the sort of exceptional species where there may be difficulties either through too few seeds or through the drying process, or through the banking, storage process, or indeed through recovery, and requiring different interventions at each of those steps. But the interventions aren't limited to the actual process of banking, but the types of material that we should focus on. And so not just for the orchids, but also for a number of recalcitrant seeded species, we've been looking at pollen storage. Now, one of the intriguing things around recalcitrant seeds, for example, in the genus Quercus or in Aesculus, is that they're highly sensitive to dehydration, but it seems that the pollen often is very tolerant of drying. And so whilst it means you perhaps could only preserve the paternal line if you're storing the pollen, we're arguing strongly that we should be flexible in thinking about using any tissue that helps you take forward the preservation of a threatened species. So that partly answers the first question again in a different way.
Thank you so much for your presentation and your answers, Dr. Pritchard. I'm afraid we're going to have to wrap things up here, given that we're quite a bit over time. And any additional questions, of course, can be sent via email.
Okay,
so
thanks.
I just wanted to thank you all for the thoughtful questions and interesting discussions. This concludes all of the invited talks and the first day of Biotech 2021. Let's give one more round of applause to all of our invited speakers today. Thank you.
All right, before we leave today, we have a few reminders. There are 41 very wonderful posters on the website covering a range of topics. So if you haven't had a chance to check them out yet, you can click on the tab labeled poster on the conference website.
Also, tomorrow we will have contributed oral presentations in 2 separate rooms. In room 1, we will have the plant molecular research session and the plant protection session in the morning. And in room 2, we will start with the micropropagation session and the physiology and production session.
For more information about the schedule, please check the conference website. Have a good rest of your day, and we will see you again tomorrow at 9 a.m. Bangkok time.
สวัสดีค่ะ
สวัสดีครับ